A very good morning to all. Today we are going to see about the complement system. This complement system will comes under the unit 3. Let us see the overview of that. We are going to study about the activation pathways and immune effects in the complement system. Complement system will have the group of 20 plasma proteins, which is essential part in the innate immunity. The word complement enhances our complements our immunity. The complement proteins will synthesis in liver, which is widely distributed among the body tissues and fluids, which comes into effect only when it get activated. 11 proteins are mostly involved out of 20 proteins named as B, C1 to C9 and D. Enzymes precursors catalyze a series of enzymatic reaction which will form the products of complement system. How is it activated? It activated in the 3 pathways. One is the antigen-antibody reaction, otherwise we called as a classical pathway. Second one is the bacterial endotoxin, otherwise known as alternative pathway. Lectin pathway or the mannose binding lectin will involved in that pathway, classical pathway. In this classical pathway, antigen-antibody reaction is the main triggering agent. This will trigger out the C1 component. That C1 will go and cleave the C4 into C4A and C4B. Now the C1 will combine with the C4B, will become the C1,4B. Again this C1 will cleave the C2 into C2A and C2B. This C2A and C1,4B will combine together C1,4B to A, which act as a C3 convertase. This C3 convertase will cleave the C3 into C3A and C3B. This C3B will combine with C1,4B to A, 3B, and it will act as a C5 convertase. This C5 convertase will cleave the C5 into C5A and C5B. This C5B will activate the other components like C6, 7, 8, 9. This C5B 6, 7, 8, 9 will act as a membrane attack complex. Now this membrane attack complex will kill your bacteria. C3A and C5A will have the other immune effects. Now the alternative pathway. For this alternative pathway, which is triggered by the bacterial endotoxin, normally this endotoxins will present in the lipopolysaccharides and the outer membrane of the gram-negative bacteria, yeast cell wall, and also the cobra venom factor. This will cause the spontaneous hydrolysis of factor C3, and it will cleave the C3 into C3A and C3B. This factor C3B will combine with factor B, and it will trigger out the factor D to cleave the factor B into BA and BB. This BB will combine with this factor C3B and acted as a C3 convertase. This C3 convertase will cleave the C3 into C3A and C3B. This C3B again combined with this factor C3B, BB and 3B act as a C5 convertase, and it will activate the other systems and it will act as a membrane attack complex. In lectin mannose pathway, mannose binding lectin MBL will be a main triggering agent in this pathway. Neisseria, Candida, Salmonella will have the mannose sugar, which is synthesized by the liver in the human being. This MBL will activate by the MASP. MASP is nothing but mannose binding lectin associated serine protease enzyme, which will cleave the protease or activate the protease enzyme, and it will combine with the MBL and it will cleave the C4 and C2 into C4A and C4B, C2 into C2A and C2B. Now this C4B and C2A will combine together C4B and C2A, which will act as a C3 convertase. Rest of the thing you know guys, which we have seen in the classical pathway. Now we are going to see the immune effects of complement products. The first product is ozonization. C3b will activate the neutrophils. That neutrophils will enhances the phagocytosis process. Next effect is cell lysis. Membrane attack complex will do the cell lysis process by the help of C5b6789, which will rupture the bacterial cell wall. Chemotaxis effect. The C5a will activate the neutrophils and macrophages, where the area of immune process will occur. The activation of mast cells by C3a, C4a, C5a, which will activate the mast cells and basophils, and that will work as the mediators of inflammation, releasing of histamine and serotonin components. Thank you.